Hello, everyone. My name is Jenny Hein, and today I'm excited to present my research on the evolution of Coxa-Trochanteral joints in beetles. Now, while my research is about beetles in general, I will focus only on weevils in my presentation today. Let me introduce myself very briefly. My name is still Jenny, and I'm currently a PhD candidate in Germany at the Karlsruhe Institute of Technology, commonly known as the KIT. And I've been working here since April 2023, focusing on the evolution of beetle morphology. Now, if I had to summarize my whole project into just one sentence, it would probably be something like this. So my main research goal is to correlate morphological data with phylogenetic trees to better understand the key events in the evolution of hip joints in beetles. Now, usually I would dedicate a couple of slides to introduce beetles and their diversity and why they are an amazing group to study. But I'm pretty sure that this audience is already aware of that, so I will jump straight into my research. Let's briefly clarify what and where these Coxa-Trochanteral joints are. So here we have two 3D models of a weevil, and the Coxa-Trochanteral joints are highlighted in red. And these joints play a vital role in the beetle's movement, of course, and they are the main point of interest in my study of beetle morphology. But why are we even interested in these joints? So a few years ago, it was discovered that the hip joints of weevils possess a very unique and special structure. The so-called screw joints consists of the coxa, shown here in green, and the trochanter, depicted here in yellow. And both of those exhibit very distinct coils that perfectly interlock, allowing a rotation of those components, which is strongly reminiscent of a screw with a nut. And this allows the weevil to have a higher degree of mobility and stability, enabling them to navigate through their environments more efficiently. I have represented this again here with my own data. So up here is again the 3D model of the weevil. And here you see the coxa-trochanteral joints up close. And this is the cross-section, so we're looking inside the joint. And to better understand why they are called screw joints, I have prepared this little animation where you can clearly see how the trochanter rotates along the coils of the coxa, similar to a nut on a screw. And in this design, where both the coxa and the trochanter have these coils and form this perfect screw and nut unit, this has only been found in weevils so far. And during my project, I will investigate the evolution of these joints. So we know that the Cocoleonidae have developed the special screw joint. However, the joint types for many of the weevil's close relatives are still unknown. And by studying these joints, I want to find out if they have the same screw-like structure as the Cocoleonidae do, or if they are different. And this will help us to understand how these joints evolved and adapted inside the Cocoleonoidea superfamily. Here's the distribution of specimens that I have been able to obtain, scan, and also segment. Now, while I have a lot of different specimens from the Cocoleonidae, most other families have been pretty difficult to get, especially the Belidae and Caridae, because I only have dried specimens for those families, which means that they are pretty badly preserved, which makes it an absolute hassle to segment. Now let me show you. Here's an example of an alcohol-fixed specimen, and outlined in red is the coxa. So you can clearly see the borders of the coxa, the muscle fibers, and also the ligaments. And here is a dried specimen. So this is also the coxa here, but it has all this excess material, which does not really belong, but it's also hard to distinguish from the true border of the coxa. So even if I segment this, it will look extremely messy, as you will see a little bit later. And I probably won't be able to use it for certain shape analysis methods. So I hopefully will be able to obtain more specimens for these families, but also the other families to have at least 2 to 3 specimens for each family. And here's the distribution of number of specimens for the different subfamilies inside the Cocoleonidae that I have got so far. And here I would also like to have at least 2 to 3 specimens for each subfamily, as well as some additional subfamilies, because I want to correlate morphology with phylogeny. So it would be amazing to look at all the subfamilies that are included in the phylogeny that I'm using, which would be the one from Shin et al. So I hope I will be able to gather most of the missing subfamilies as well. So now you have an overview over the specimen that I want to use to reconstruct the evolution of the screw joint inside the Cocoleonoidea. And I will do that with the help of high throughput X-ray microtomography. So first of all, of course, I needed to obtain the beetles, a lot of beetles, for which I have been able to obtain about 300 species so far. Not just weevils, but all kinds of beetles, either by collecting them on my own or from Malaise traps or from the collections of the Natural History Museum. And then these beetles were scanned at the KIT light source using high throughput tomography, and the reconstructed scans that look something like this, they can then be used by me to segment the structures that are interesting so that in the end I have a 3D model of this structure. In this case, you can see the whole segmented beetle here, but I would only focus on the middle hip joints, which would be the area inside this red circle. But I actually don't segment every beetle manually because it would just take way too much time to do it for 300 specimens. But instead, I use Biomedisa, which is a segmentation tool that uses a few pre-segmented reference slices to automatically and accurately extend the segmentation across the entire 3D image. And to gain deeper insights into how these joints function, we are also utilizing 3D printing. And by creating physical models of these joints, we are able to study their movement in a more tangible way. And this allows us to observe how the different parts interact realistically, which can sometimes be challenging to fully capture in digital models alone. And here you see some of the joints that I have been able to print already. Now, here we see the morphological diversity of the trochanter among various families within the Cocoleonoidea. And each model represents the trochanter morphology from a different family, with the most primitive, the Cimberellidae, on this end, and the most derived family, Cocoleonidae, on this end of the tree. Now, the question was if the closely related families of the Cocoleonidae also show the same type of joint morphology. And I really hope that you agree with me here that these trochanter look pretty different on the first glance. And the reason why I only show the trochanter is because the coxa morphology might also vary strongly between different families, but it also varies strongly for species of the same family. If we just look at a few different species of the Cocoleonidae, for example, we see very different coxa morphologies, even for closely related species. On the other hand, the morphology of the trochanter is still pretty similar among all species. And this can also be observed for other families. So here we see some more species from the family Brentidae, which are the closest relatives of the weevils. Now, the coxa morphology is not as different, but still the morphology of the trochanter is pretty similar among the species of the same family. And I could observe this trend for every other family inside the Cocoleonoidea that I had multiple species of. Okay, now I have all this data and all these models. So what I will work on next is first of all, morphological descriptions of the joints for the different families. Then I want to use some geometric morphometrical methods to analyze the joints. For example, using landmarks to compare the coils of different screws, different screw joints. But I'm also looking into landmark-free approaches right now. And to get a better idea about the ecological advantages that the different joint types might bring, I want to correlate the joints with different parameters and hope to find some correlations there. And lastly, we are going to build an open online database that contains full body scans of all the beetles that I'm working with, not just weevils. And we plan on also barcoding all specimens to then also include the genetic information in this database. So with this, I'm at the end of my presentation. Thank you very much for listening, and I will be glad to answer any questions.